Today my topic is using RNAseq for evaluation, safety evaluation for oligonucleotide therapeutics. But I think most of you are not familiar with these tools, since we are just staying in the more comfortable way. When you get this kind of big data, so maybe you are not comfortable with this. Okay, actually this is a KIT sponsored session, so I just introduce our institute in brief. So KIT is a government-funded research organization, and it founded in 1982. Is the name of Toxicology Research Center. But we are growing up, and in 2002 we started as a KIT. So now we have 3 campuses around the country, South Korea. The headquarters is located in Daejeon. We are doing general tox, repro tox, safety pharmacology, and genetic toxicology in Daejeon. And then R&D department is also in Daejeon. And then we have another campus in Jeongup. We have an inhalation toxicology facility, and then we are doing also doing general toxicology. But in Jeongup, we have a monkey facility. So their its capability is like 600 monkeys in the house. Also, we are doing the 2 years carcinogenesis study in Jeongup. And then we have another campus in Jinju, the southern part of Korea. They are especially doing ecotox and then doing some K-REACH on EU-REACH service. So this is our department. So we have 3 research centers. One is the systemic toxicology, and then another is the next generation pharmaceutical research center, and then another one is alternative toxicology research center. We are 34 people work in our department, 17 PhD. So this is about 10% of our whole institute. So our ultimate goal is using this kind of the model and technology. So finally, we wanted to predict toxicity in drug or some other things. Okay, this is contents for my presentation. So we are saying next generation sequencing, NGS, is a new method for sequencing genome in a high speed and low cost and with great accuracy. So the first generation sequencing method is based on Sanger, but this is the second generation. So we are doing more speedy and then low cost. So maybe you already know this graph. So in early of 2000s, the Human Genome Project was done, and then at that time, it's a Human Genome Project. It costed like $3 billion. It took 10 years for the completion, but you can see there the cost per genome is dramatically decreased when the new technology was developed. So this is one of our benefit with technology. So actually, when I started my postdoc, at that time there's the phrase like one gene, one postdoc. But now it's changed, whole genome with one postdoc. So this technology took away many postdoc job position. Anyway, but when you get the tons of data from the high technology tools, so you maybe embarrassed with the amount of data. So this is a very brief scheme for when you doing with this kind of new technology. So sample preparation, and then the sequencing, and then image analysis. Usually it takes 2 or 3 weeks for sample publication, but it actually is the mathematical calculation. Actually, I'm a kind of the challenging person. So I'm majoring doing with the chemical drug. But recently, I was interested in this kind of the new tools for the oligo. So we check out the RNAseq. Actually, the RNAseq is they count the sequence. It means transcript abundance. It's the qualitative and the quantitative. So it looks like the micro give out a lot of data like a microarray, and then it also give out high resolution like the qPCR. And then this is not from my story, but it's appeared on Nature Review 2009. So there's a comparison table for this technology. Maybe you know about the tiling microarray and the cDNA or ESD sequencing, and then RNA-seq is here. So they said it's compared to tiling microarray, it has a very low background. So it's very more dynamic range for gene expression level, and then it's very low cost. So there's many advantages with the RNA-seq. So also we checked the recent publication with this RNA-seq for the oligo development. It's quite a recent paper. So this kind of the disease-based work done by RNA-seq, but safety matter is not done with this new technology. So we built up the RNA-seq analysis pipeline recently. So when you get the raw data from NGS, maybe you investing with this kind of the big things, but and then also NGS give a result from this reference genome mapping. So this very small part is like the sequence like when you shoot a shotgun. So they made the full sequence of RNA. So recently we built up this kind of the analysis facility in KIT. So we are building up a more computing part in the future. So when you done with this RNA-seq experiment, maybe you get more than giga. It's based on terabyte of amount of data. So actually, we are doing some collaboration with the university for this kind of the RNA-seq things. So it's chemical-based toxicant and or the ES cell drive the cardiomyocyte the RNA sequencing. Also the inflammation inhibitory study with the RNA-seq. Also we are doing the zebrafish RNA-seq. These things are all based on the chemical drug. So we analyze RNA-seq data with this flow, and then we get finally we can get a DEG or some other networking and the pathway from the gene information. Also, we built up the protein interaction analysis. So in the future we can get more information with the network from the gene expression. So as I mentioned previously, I'm majorly working with the chemical drug. So this is our first approach using the oligo for RNAseq. So we got some sample from our collaborator. It was done by our facility. The mouse was is a C57 black 6 mouse. So the whole treatment duration like 7 weeks, and yeah. So in that during the treatment, some of the group animal group they found that or the moribund status was observed. So we took liver sample from the mouse, and then we did the RNA-seq with the liver sample. So in group 3, OT2, their group, so most of the animals show the found death and then the moribund status. So in this case, we check up the RNA integrity. So they show the RIN value like the 5. So when you doing with the RNA, so the RIN number 5 is like we cannot use in the microarray. But as I mentioned before, I'm a challenging person. So we try to with the RIN number 5 for in this case. Okay, this is the organ weight or clinical chemistry data from the mouse. But in OT2 sample, so we can get like 73% of the mapping of all our sample. So I think the RIN number 5 is still okay with the RNA-seq. So some case we get sample from patient from hospital. In the case, the RIN number is very low. But I think that we have some possibility with the low RIN number can make a relatively good result from RNA-seq. So we got the gene number, the differentially expressed with expressed gene. So compared to other groups, the OT2 sample showed very large number of gene expressed in the upregulated. So for our analysis, we just excluded OT2 when we doing the analysis, since there's maybe there's some other fact in the OT2. So we the group the OT1 and OT3, OT4 is in the same group, and the OT2 just excluded for the analysis. So this is the one picture for when we're doing the RNA-seq. So we can check the target gene part. So this is more the big picture with this part. So this is a target gene for with this the oligo. So control shows that there's an intact status, but OT2, they affect of many parts of the target gene area. Then OT3 is relatively mild, but it's also affected with the oligo in the target region. So also RNAseq shows the result with the digital number. So actually this number means the gene read count. So compared to control, in the case, the OT2 and OT3, they decreased in target number region. So we can get it's a digitalized number with RNA-seq. Okay, this is the up-regulated gene. As here, it's like a co-up-regulated gene in moribund state. We just excluded the OT2 group. So you can see that so many hepatic fibrosis factor, and then hepatic stellate cell adhesion, and the IL-17, IL-10, LPS-related gene was up-regulated with treatment. So with this, and then we can pick up some of the pathway in the inflammation stage. So you can get this kind of information from our result. So this is just a part of our analysis. So if you want, if you are interested in more part of the other up or down regulated region, you can get this kind of picture. And then so which part was affected with treatment. And then this is the down-regulated gene in the moribund status animal. So usually the complement is a very big issue when we treated oligo. So in this analysis, we get down-regulated complement system. But usually the complement is up-regulated with the oligo treatment. So we also check up the complement system. So in this analysis, the oligo just affect on this classical pathway of complement system. The alternative pathway was intact, but this oligo just down-regulated this part of the complement system. So as you can see, this is very, with this data, we can get more specific reaction in the biological system. So maybe we can get more the clue for the most safe oligo drug. Okay, this going to be less data for RNA-seq. So we just check that OT2 upregulation. It showed 3 or 4 times more higher gene number expression. So in OT2, they show the high score in the diapedesis in their upregulated gene. So I think that I'm not an expert in oligo, but diapedesis is also the big issue in the toxicology of oligo. So this is the part of our analysis, but you can get more many things from this kind of RNA-seq data. So yeah, so we can get more genome-wide scale data for the efficacy and the toxicity profile of oligodrug candidate. Also, RNAseq can get data sequence independent toxicity profile, like the hepatic fibrosis, toll-like receptor signaling, and other oxidative stress pathway. So, so this is more, you can get more comprehensive analysis with this RNA-seq data. So this is the very limited data for the future, but I think this kind of approach can make more useful things for early development of oligo. So for the study, I just suggest for this in the future, for the future. So actually, we are doing stem cell research. So we are doing from the embryonic human embryonic stem cell or human induced pluripotent stem cell. So we are deriving hepatocyte, cardiomyocyte, the neural cell from the human ES or human iPS. So this is the part of our the government grant. So we are doing this for the human-like, human cell-like hepatocyte and cardiomyocyte and neuronal cell for the direct drug development. But I think we can also apply this kind of cell for the oligo. Since we get very good quality of hepatocyte from our project, so this is the picture for the human ES cell-derived hepatocyte. It shows very good characterization of human hepatocyte. So usually we are doing in vitro assay with human carcinoma cell line, but I think that's not enough for the in vitro screening. So this is just I think this is more challenging work for me. But when we using this kind of the human-derived hepatocyte, so maybe it can get more reliable data for in vitro study. Since we got the result in mouse, actually it was some screening study in vitro with this kind of such kind of the human carcinoma cell line. So maybe this kind of ES cell derived hepatocyte make it more helpful for the in vitro study. Okay, so I also thanks to my collaborator for bioinformatics and the stem cell group and then non-clinical study group in Changok. Thank you.